Welcome to this explainer. Let's jump right in, because today we are looking at an absolutely fascinating intersection of nature and technology. We're breaking down a major 2024 breakthrough by Manochkumar, Jonnalagadda, Cherukuri, Vannier, Janjaroen, Chandrasekaran, and Ramamoorthy. What they've done is, frankly, incredible. They figured out how machine learning prediction models can unleash the enhanced production of a highly valuable marine pigment called Fucoxanthin right inside a specific microalga known as Isochrysis galbana. It's basically a masterclass in how data science is stepping in to solve some of the most complex bottlenecks in biotechnology.
Now, to really grasp why this matters, you have to look at this number. If you want purified fucoxanthin today, you're looking at anywhere from $40,000 to $80,000 for a single kilogram. I mean, that is a staggering price tag. We are literally talking about liquid gold here, and the global demand is desperate for it. So you might be wondering, why is it so outrageously expensive, and why haven't we just figured out a way to mass produce it cheaply in a lab? Well, that is exactly the puzzle this research solves.
So, here is our game plan for today. We'll start with the golden marine pigment, move into the microalgae factory bottleneck, enter machine learning models, test the magic ingredients, reveal the winning combination, and finally, look at the future of sustainable biotech.
Let's kick things off with part 1, the golden marine pigment, a biological treasure.
Okay, so fucoxanthin is a marine carotenoid. In nature, it sits inside the chloroplasts of algae, acting sort of like a tiny solar panel, absorbing light for photosynthesis. But for us humans, it is an absolute powerhouse. It offers immense health benefits, showing real promise in preventing chronic conditions like cancer, cardiovascular disease, and diabetes. Because of this, the market is just exploding, and it's actually expected to hit almost $281 million by 2029. But here's the catch. You cannot make this synthetically right now. The molecular structure is just way too complex. So we have to rely entirely on nature. Specifically, a microalga called Isochrysis galbana. We use this one because it lacks a rigid cell wall, which makes extracting the pigment a lot easier, and it grows pretty fast. But getting it to reliably produce enough of this pigment on an industrial scale, that is where we hit a massive wall.
Which brings us perfectly to part 2, the microalgae bottleneck, industrial roadblocks.
The actual roadblock holding everything back isn't just growing the algae itself, it's measuring what they produce. The traditional method is called HPLC, or high-performance liquid chromatography. Think of it like trying to run a fast-paced commercial kitchen, but the health inspector takes over 3 hours to test every single dish before it goes out. It's tedious, it's incredibly expensive, and worse, it uses toxic, hazardous solvents like acetylnitrile and methanol. It's just not sustainable. But the new method proposed by this research team changes the game completely. By combining UV spectrometry with machine learning, testing takes just 5 minutes. 5 minutes! It uses minimal, far less toxic solvents like ethanol and remains highly accurate. It literally shifts the entire industry from a slow, toxic slog to a hyper-efficient, sustainable process.
And this setup brilliantly illustrates the workflow they designed to smash through that bottleneck. You've got the biological side, where they scale up the Isochrysis galbana and treat them with varying concentrations of plant hormones. Then, that rapid extraction and UV spectrometry analysis happens. All of that raw biological data, the growth rates, the biomass, the pigment yields, it flows directly into the data pre-processing stage. And finally, that cleaned-up data gets fed right into various machine learning algorithms to evaluate and predict the ultimate fucoxanthin yield. It's just a beautiful marriage of wet lab biology and high-tech data science.
All right, part 3. Enter machine learning, predicting yields.
I like to think of these 4 machine learning models as 4 master chefs, all competing in a kitchen to figure out the absolute perfect recipe to make our microalgae produce the most pigment. First up, we have random forest. This uses ensemble learning, meaning it consults a whole forest of decision trees to make a really robust prediction without jumping to wild conclusions or overfitting the data. Then we have the support vector machine, which is amazing at drawing precise boundaries in highly complex, multi-dimensional data spaces. We've also got linear regression, the classic approach, looking for straight-line mathematical relationships between the ingredients and the final yield. And finally, the artificial neural network. This one mimics the human brain, designed to recognize those deeply complex, hidden, nonlinear patterns that the others might miss. The researchers essentially pitted these 4 algorithms against each other.
Moving on to part 4, testing the magic ingredients, phytohormones.
So, the crucial biological element here is that certain plant hormones physically alter how these microalgae grow, Looking at these field emission scanning electron microscope, or FESEM scans, you're seeing the actual surface of these cells. In the control group, you see nice, clustered, relatively smooth cells. But watch what happens when we introduce hormones like IAA, salicylic acid, gibberellin, and specifically methyljesmonate. The cells physically change. When treated with low concentrations of methyljesmonate, they become beautifully swollen and enlarged. That is exactly where maximum pigment production happens. But if you push it too far and overdose them at, say, 20 milligrams per liter, the cells become distorted, way too swollen, and essentially just explode. So the AI's entire job is to find that exact, perfect sweet spot before the cells burst. They also use confocal laser scanning microscopy to peer directly inside the cells, and the colors here tell an amazing story. That red glow? That's the natural autofluorescence of the chlorophyll. The green speckles represent the lipids, and most importantly, our golden target, the carotenoid pigments. By analyzing these scans, the researchers could literally watch it happen. While some hormones just destroyed the red chlorophyll, one very specific dose of methyljesmonate, exactly 0.2 milligrams per liter, triggered this massive explosion in green fluorescence. The cells were practically pumping out the fucoxanthin.
Let's look at Part 5, the winning combination, the ultimate recipe.
Now what's really fascinating is how the AI actually views all of this. This is a Pearson's correlation heat map. It might just look like a colorful, complex quilt, but it maps out incredibly intricate relationships. You see, the researchers didn't just tell the AI, hey, we used hormone X. No, they gave the AI the deep chemical descriptors of the hormones. We're talking hydrogen bond donor counts, topological polar surface area, and pure molecular complexity. The deep red squares highlight strong positive correlations. By feeding the AI the underlying chemical traits rather than just the surface-level biological outputs, the models were able to spot incredibly subtle patterns that a human researcher would just completely miss.
And once all that highly complex chemical data was processed, we had a very clear winner. The random forest model absolutely crushed the competition. When those intricate hormone descriptors were included in the mix, random forest achieved an incredibly high accuracy score, with an R-squared of 0.839. The artificial neural network came in a solid second, while the simpler models, like linear regression, really struggled to capture the messy reality of biology. The random forest proved that its ensemble learning approach was perfect for filtering out the biological noise to find the true, actionable signal.
So, drumroll please, here is the winning formula predicted by the AI and verified in the lab. Supplementing the microalgae with methyljesmonate at a concentration of exactly 0.2 milligrams per liter produces maximum fucoxanthin in under 10 days. Just to put that into perspective, a normal control group would take up to 18 days to reach peak production, and even then the yield would be significantly lower. This prediction is just a massive leap forward. It eliminates endless biological trial and error, saving so much time, money, and resources. They've effectively taken a chaotic natural biological process and turned it into a highly predictable, optimized manufacturing line.
Finally, let's wrap up with Part 6, Sustainable Biotech, Blueprint for the Future.
What you should really take away from this explainer is that this isn't just about one single pigment. It is a blueprint for the entire future of biotech. First, it proves without a doubt that AI unlocks rapid, data-driven discovery, ending the era of blind trial and error. Second, right now, most fucoxanthin is harvested from macroalgae, or seaweed, which can actually severely disrupt delicate marine ecosystems. By optimizing indoor sustainable microalgae farming instead, we actively protect our oceans. Third, it proves machine learning's immense scalability. If it can clear a bottleneck for this specific compound, it can do it across all of industrial biotechnology. Ultimately, this exact integration of rapid chemical scanning and AI lays the groundwork for how we are going to manufacture the next generation of drugs and cosmetic ingredients. It is data and nature finally working perfectly together. It truly makes you wonder, doesn't it? We've just seen how giving an AI the underlying chemical blueprint of a plant hormone allowed it to perfectly optimize a microscopic marine factory. We literally bridge the gap between data science and marine biology to easily harvest a compound that helps fight cancer and heart disease. So, I'll leave you with this thought. If machine learning can unlock the secrets of microalgae today, what incredible biological mysteries will it solve tomorrow? Thank you so much for joining me for this explainer. Keep learning, and I'll see you next time.